Welcome everyone to this NanoFutures webinar on seamless integration of functional nanomaterials and nanoelectronics with biology. This is based on the article recently published in NanoFutures by Professor Jia Liu and co-workers with the title Functional Nanomaterial Enabled Synthetic Biology. You can read the full article on the journal homepage. Joining us today will be Professor Bozi Tian from University of Chicago, who is on the editorial board of NanoFutures and chair of today's webinar, and Professor Jia Liu from Harvard University. Professor Liu's talk, we will have a question and answer session. Please use the Slido link to submit your question to our speakers. You can access the webinar Slido using our code NanoF2021, and the link to the Slido is being added to the chat now for you to access. I will now hand it over to Professor Tian, who will be introducing the journal and our speaker. Professor Tian received his Bachelor and Master of Sciences degrees in chemistry from Fudan University, Shanghai, in China, and his master PhD degrees in physical chemistry from Harvard University in 2010. He's currently a professor in the Department of Chemistry at the University of Chicago, and his current research focuses on the semiconductor-enabled understanding of subcellular biophysics, as well as studies of dynamics at soft-hard interfaces. Over to you, Bozi. Thank you.
Well, thank you very much for your nice introduction. Hello, everyone. Welcome to the NanoFutures webinar. My name is Bozi Tian, professor of chemistry at the University of Chicago. Today, I'm very honored to host this NanoFutures webinar given by Professor Jia Liu from Harvard University. Before I introduce Professor Liu, I would like to just highlight the NanoFutures journal. This NanoFutures is a multidisciplinary and high-impact journal publishing both the fundamental and also applied research at the forefront of nanoscience and technological innovation. The current editor-in-chief is Professor Amanda Burnett from Australia National University, and who is also a theoretical physicist. So I'm a board member, and I have been working with my other colleagues over the past few years. So this journal was launched in 2017, and so far it goes very rapidly. It publishes papers with diverse topics in nanoscience and nanotechnology. So our speaker today and his group has recently published a review paper on this webinar topic in the NanoFutures. So now let me introduce our speaker today, Professor Jia Liu. So Professor Liu is currently an assistant professor at the School of Engineering and Applied Sciences at Harvard University. Professor Liu and we did our undergraduate studies in the same lab in China, although we did not have any overlap back to that time. Then at Harvard, Professor Liu and I had a few years of overlap when we did our PhD. And since then, we have long-term connections. So Professor Jia Liu has developed a variety of tools that combine nanoelectronics, polymer chemistry, genetic engineering, and tissue engineering to address multiple challenges in neuroscience, neuroengineering, brain-machine interface, and cardiac arrhythmia. So his work has established some of the fundamental principles for the current state-of-the-art tissue and brain-machine interface used in both the academic research and also industrial applications. For example, Jia has developed an approach to the direct formation of electrical connections with neurons via the genetically targeted synthesis of conductive polymers through the modified surface proteins. And this work has completely changed the way people think about neuroscience and neuroengineering. And from my view, this work represents probably the most exciting advance in bioelectronics over the past 10 years. His lab at Harvard University is primarily focused on the development of new bioelectronic and cyborg engineering, genetic and genomic engineering, and computation tools for biological and biomedical applications. He also co-founded and served as the scientific advisor of Exosoft, a brain-machine interface company. And as an independent PI at Harvard University, Professor Liu has received numerous awards and honors, and I won't list all of them. I will just pick a few. For example, the most recent NIH and NIDDK Catalyst Award. This is the DP1 Director's Pioneer Award program. That's probably the most prestigious one from award from NIH. And he also got award from MRS, like the MRS Best Symposium Presentation Award, and also many awards from Harvard University. Yeah, so with this, I would like to welcome our speaker today, Professor Jia Liu. Jia, the floor is yours. Thank you very much for coming to us.
Thank you very much for your nice introduction, Bo-Zhi. And it is my great pleasure to present our recent work in this NanoFutures webinar. And we recently published our review paper on the NanoFutures. So feel free to look at the paper after this presentation. So it is my great pleasure to present you some of our very recent progress in the lab that about how to make this seamless integration of the functional nanomaterials and the nanoelectronics with biology. So my group at Harvard are interested in those questions such as how the individual neurons function together to give rise to behavior, or how those like malfunction of the cells that give rise to the different kind of the disease. And the last one is how those like cells like organize together through a self-assembled way to grow into a piece of the organ. And to be more specific, we are very interested in building those next generation bioelectronics to understand those questions. Specifically, we wanted to convert those like high performance nanoelectronics fabricate on the silicon wafer into a soft bioelectronics format for this tissue machine or brain machine interface to address those questions. So before I talk about the device that we are developing, I want to firstly like highlight those like challenges that we try to address to build the effective brain machine or tissue machine interface. The first one is that if we use a brain as an example, so when we want to start its functions, those single cell activity across the entire brain really matters. So unlike our computer chip, brain function in a complete different way that when that the cells like distributed across the entire 3D volume of the brain need to function together to give rise to certain behavior. So this picture on the left, that is the functional MRI imaging of a human patient. And those like red colors label the neurons that have this activity. So you can see that under certain brain internal state or behavior state, there are neurons that are distributed across this 3D volume of the brain that show the activity. So this picture on the right, that is the calcium imaging of a zebrafish brain. Zebrafish is a transparent animal, which the entire brain is transparent. So those red dots highlight the neuron that is firing. So you can see that for the certain behavior or internal brain state of the zebrafish, they involve the neurons that are really distributed across this 3D volume of the brain to collectively give rise to that behavior. So if we want to really understand those brain functions, ideally we want to achieve this single cell level recording across the entire 3D volume of the brain. But if we want to achieve this high level of the recording, then we will face the second challenges, which is called the spatial temporal dilemma in neuroengineering. So consider those like a neural activity, it operate at the millisecond temporal resolution. So the one that in the middle, that is the action potential of one neuron, which is have the duration around like 1 to 3 milliseconds. However, we know that the neuron and the brain activity are not operating at the millisecond temporal resolution. It involves like it takes days, months, or even years to build up the emotion, cognition, or the learning. And meanwhile, those individual activity that is confined into the individual neurons, which have the spatial dimension from the 10 micrometer to 100 micrometer. However, if we involve the neurons distributed across the 3D brain, that is at the spatial scale from the centimeter or even to the meters. So in addition to consider, there are a large number of the neurons in the animal and in the human brain, such as millions of the neurons for the frog or billions of the neurons for the human. We basically want to achieve like at least like a 500 gigabytes per second or even thousands of terabytes per second to capture this large number of the activity across the 3D brain. And to give you a comparison, the 720p movie that you can watch on the YouTube, that is only 2.5 megabytes per second. So there is a huge gap between what we can do right now to what we want to do in the future to build this large bandwidth for the brain recording. But why we think this is time that we can do that? So on one side, this is the picture on the left that is driven by the Moore's law. The number of the transistors that integrate on individual chip already equally to or even beyond than the number of the neurons in the human brain. And on the right, that is due to those like progress in nanotechnology, we now can make the miniaturized electrical detectors and the stimulators, and in the in vitro setting can probe the neurons in a very long-term stable way at the subcellular spatial info resolution. So if I put those like two examples here, on the left, that is the human brain, the wiring of the billions of the neuron, and on the right, that is a standard chip that is also billions of the transistor into that. So we have the number right now. We will very soon we will have the number of the transistor in a single chip that is beyond the number of the neuron in the human brain. And each transistor can operate at the speed that is much faster than the human brain. So what we are really missing here, that is a cable technology, an interfacing technology that can bridge those individual electrochemical impulses that operate in the individual neurons from this 3D brain to the hardware, to the powerful chips that we have to interpret or collect their information. So to build this kind of like an interface, since we have those like large number of the neurons that we need to address, so what we need is actually a scalable technology for those like brain-machine interface. So this diagram basically summarizes the need for this type of the scalable technology. The X, that is the spatial, that is temporal scale, and the Y, that is the spatial scale. And this two dashed line basically highlight where we call the single cell and the single spike spatial temporal resolution. And so this is how the like a traditional microelectronics or bioelectronics that can do. This red and the blue lines basically label their spatial temporal resolution, and this pink area that is highlighted the like a spatial temporal scale that they can cover. You can see that for the traditional bioelectronics, they can easily achieve this single cell, single spike spatial temporal resolution. However, they cannot last for a very long time in the brain. And of course, they cannot be easily distributed across the entire 3D of the brain. So the first part of my talk is that we try to understand how to address those spatial temporal dilemma. So there's one direct solution that can we make, for example, the like a brain probe, like those like a computer chip, using those CMOS technology to build the large number of the sensors and get into the brain to record the neural activity? Can this really solve the problem? So someone already have done this by developing this very beautiful, very elegant CMOS-driven multiplexed electro array that is called the neural pixel, and can simultaneously look at the hundreds of the neural activity in the brain. However, you can see that over the time, the number of the neurons or the quality of the neurons will be rapidly decayed over the time course of the implantation. They can still not stably record the neural activity inside the brain for a very long time. So the first part of my talk, I would like to introduce you the concept to build the tissue-like nanoelectronics that to provide this long-term stable brain-machine interface at the single cell level to substantially extend the spatial scale that the bioelectronics can record the brain activity. So to address those questions, we first need to understand what's the limitations, what's the challenges that prevent us from achieving this long-term stable electrical recording, this long-term stable tissue electronics interface. So consider brain is very soft. It is like a tofu or like a pudding. And conventional electronics is very rigid, like a blade. So if you put the electronics into the brain, it's like put a blade into a tofu. Any small movement of this tofu can cause the electronics directly cut the brain at the micrometer scale. So over the time, there is a mechanical damage to the brain, and there is also probe drift inside the brain, so that you will not be able to record the neuron stably. And in addition, due to this mechanical mismatch between the electronics and also the brain tissue, there's a large pressure from those rigid probe to the soft brain tissue, which will also trigger the foreign body effect, causing the proliferation of the microglia and astrocytes, those immune cells in the brain tissue, which isolate the probe from the healthy neurons. So over the time, you will lose the capability to record the neural activity inside the brain tissue. So here shows a picture of a brain tissue after the before and after the long-term implantation. And in the middle, the cavity that is the implantation site, the green color label the neurons. You can see that over the time course of the implantation, there was degradation of the neuron at the implantation site, which will form like a 200 micron, 50 to 200 micrometer so-called neuron killing zone. And the red color labels the astrocytes, one type of the immune cell. You can see that over the time, there's a proliferation of those immune cells around this implanted probe, further isolate them from the neuron. So you will quickly lose those single unit action potential that you can record in the very beginning. And for the patient with deep brain stimulator implanted, every couple of the months or a year, you need to relocate the stimulators to find the healthy neuron to do the effective stimulation. So in the first part of our research, we really want to ask the question, how to design the electronics that can allow the seamless integration with this 3D brain tissue, which can allow us to integrate the electronics across multiple and including the deep brain regions, and completely eliminating the glia response and the neural death to provide the stable chronic electrical recording at a single cell and a single spike spatial temporal resolution. So to solve this problem, we really want to look at the history of the biomaterial. Is there any material that can provide this kind of gliosis-free implantation in the human body? Actually, people already developed this type of material, which is the so-called synthetic tissue scaffold that has been already widely used for the regenerative medicine. And those material is a 3D microporous soft biomaterial, which can allow the cell grow inside of them, form the connection, and ultimately into a 3D piece of the tissue without any cell degradation damage or the immune response. So the first part, we want to develop the nanoelectronics to mimic this structure. We think the nanoelectronics should have this 3D microporous structure with subcellular feature size. And most importantly, they need to have the tissue level mechanical property, like the softness of the flexibility. And in fact, all the nanoelectronics need to also be fabricated through the photolithography or even lithography fabrication method, so that we can leverage this a semiconductor industry level fabrication capability to highly integrate a large number of the sensors and the stimulators into those bioelectronics. So the idea is very simple. So this is a work which is done by me and Bo, which is almost 10 years ago, that because the development of the nanoelectronics, so there's so many nanoelectronics components that can be freely patterned onto the different kind of substrate. We think that we can just pattern them into the soft interconnects, form like a mesh-like nanoelectronics network, and then integrate them together with the tissue to form like a 3D tissue electronics integrated structure. But the question is that how to make those soft interconnections. After all, they are occupy more than 99% volume of the device. And in fact, can we really make the soft interconnects? The question is no. Actually, we cannot make the soft interconnects like as soft as the tissue, because for all those like electronics material such as SU8, polyimide, gold, silicon, which have the Young's modulus, that is at least 6 orders of magnitude higher than the Young's modulus of the brain and heart tissues. And there may be some material like the PDMS or ICBS, those elastomer, conventional elastomers, which have a Young's modulus that is close to the tissue network. However, they are not compatible with the nanolithography fabrications. So that in the very beginning, we don't have the materials that is available for us to fabricate the tissue level soft interconnects. So the solution that we use to solve those problems is actually we can first build the tissue level flexible electronics, because the flexibility is not just like determined by the Young's modulus, it's also determined by the geometry of the objects, like the thickness. So this like bending stiffness is the parameter quantify the flexibility of an object. You can see that it is proportional to the Young's modulus, but also to the cubic of the thickness. So that in the first place, then we can make a very thin nanoelectronics to bring those like rigid electronics into the tissue flexible regime to interface with the biological system. In addition, if we want to consider about the bending energy between the electronics and the cell, we will also need to consider that we want to make the bending energy of the device features, those smallest features that is comparable with the bending energies of the cell. So put these two into consideration, and also consider that we need to have this microporous like or mesh like structure. We will have this like dimensions determined for the bioelectronics. You can see that if we consider about the bending stiffness, the device thickness need to be lower than the 5 micrometer. And if we consider the bending energy, the device thickness need to below the 500 nanometer, which is already approach to the limitations of the nanofabrication, because for all those nanoelectronics, you need to have enough thick dielectric the encapsulation layer prevent the current leak from the electronics to the biological system. So we make those structure with like a 800 to 1 micrometer thick dielectric layer. We fabricate those ribbon for the nanoelectronics, and we measure their mechanical property by using the AFM. You can see that they indeed have the bending stiffness that is equally to or even smaller than the bending stiffness of one single cell. So by making those structure, we can first part make the mesh electronics that is as flexible as the brain tissue, which can be completely removed from the rigid silicon substrate, and that is so soft, which we can deliver them into the brain like the hydrogel through a syringe-injectable manner. So this is the first example that we demonstrated in 2015, that we build those syringe-injectable mesh nanoelectronics for the brain implantation. We can inject the sensor region inside the brain, withdraw the needle, and eject those input and output pad on the outside of the brain. And after that, we can bond those like electrode to the flexible cable put on the animal for the electrical recording. However, you can see that those syringe-injectable is not a very controllable way, so that all the device may be like a bundle together inside the brain tissue, which will not provide this like optimized interface for the brain tissue for the long-term stable recording. So recently, we developed like a new method that we integrate the ultra-thin SU8 shuttles, it's like a polymer shuttles with those like mesh nanoelectronics. And my postdoc designed a very elegant way that we put some anchors so that we can incorporate a water-soluble layer between those like mesh electronics and the like SU8 shuttles. And before the implantation, we remove the anchor, we do the implantation, the shuttle can then be released from the device and withdraw from the needle. And this is showing that we test that in the transparent hydrogel, which shows the same mechanical property as the brain tissue. You can see that after the implantation, the mesh device can maintain this unfold structure inside the hydrogel to maintain this like tissue level flexible and macroporous structure with the brain tissue. So this is the example that we're showing that we can implant the like a mesh electronics with like thousands of the sensor array across this like a 3D volume of the brain across like a different region of the brain from the cortex to through this hippocampus to the different region. Those red color, that is the mesh nanoelectronics, the purple and the green color, that is the immune cell and also the neuron. You can see that there's a vanished small damage to the neuron, and also there's no proliferation of the immune cells around the probe. And we test that over the year-long recording and compared it with even other state-of-the-art thin film brain electronics, that this unfolded mesh nanoelectronics show almost no immune response and degradation of the neuron after 6 weeks of the implantation. And over this year-long implantation, there's still no proliferation of these immune cells or any degradation of the neuron. So using those like mesh electronics, we can record the brain activity across the multiple brain regions to capture this like a region-specific electrical property. And the most striking result is that we can even capture the same neural activity over the entire adult life of a behaving mice. So this is the continuous recording over 14 months of the implantation from the mice, which is at 5 months old to the 18 months old until its natural death. And the waveform on the right, each color indicates the single spike activity collected from one neuron. You can see that over this like 14 months recording, the waveform does not change at all. And the figures on the left, that is the UMAP, one kind of dimension reduction method to look at the variation of those spikes. You can see that they show the remarkable line of those like clusters over this 14 months of the recording, indicate the stability of the recording. And of course, we look at the different kind of parameters through a very intense statistical manner, like the likelihood ratio, Schleholt score, look at their stability of the amplitude, signal to noise ratio, PT ratio, recovery slope, all indicate that those like parameters have no statistically significant change over this 14 months of the recording, indicated the long-term stable recording, not at the single cell resolution, but really from the same neurons over the time. And we also do this kind of behavior related like recording. We do the whisker stimulation, and then there's one sensor that is at this like a D2 whiskers. You can see that over this 14 months of the implantation, they can keep show the response to this D2 whisker stimulation. And importantly, if we stimulate another whisker, like a C3 whisker, which is close to this electrode, but there's still no like a response. And if we record the activity from another electrode that is close to this first electrode, but away from this D2 whisker related neurons, there's also no like a response over this like 14 months of the recording. So this is very stable, long-term stable recording really gave us the capability to look at the neural activity evolution over this time course of the aging process for the animals, that we can capture, we can easily cluster and identify two types of neuron. One shows like almost no change in terms of their electrical property over the aging, and another show very clear like evolution of their electrical property, well, very subtle, which cannot be captured by those dimension reduction method. But if we zoom in, we can show that in the machine learning enabled pseudo time calculation, there is like a drift of those electrical property over the time course of the aging. So I hope that those studies convince you that we can build a tissue like nanoelectronics, which have this micropores structure, tissue level flexibility, which can provide this long-term stable recording, which substantially extend the spatial, the temporal coverage on this a diagram from the days to the years. So the next question is that how can we make the whole tissue level interface still maintain this long-term stable manner, but also cover as large volume of the tissue as possible. So one direction that is that we can do is that we can engineer in the large number of the sensors with high density electrical array get into the brain, do the whole brain level recording with a high spatial temporal resolution, which is recently started by Elon Musk, adopt this tissue level flexible electrode array that we previously developed, combined with this automatic robotics to do this large number of the recording. And we also pursue in this direction in the company, in the Xsoft that I co-founded. But this is not the key point that we want to discuss today, because there's still a fundamental limitations for this method. That is, this is a very nice EM picture that is made by the Professor Jeff Lichtman at Harvard that look at those cellular structure at the nanometer resolution. Basically, he found that all the neurons are innervated at the nanometer level. So for a brain which is already fully developed into a mature state, those neurons are highly interwoven together at the around 10 to 20 nanometer resolution, so that no matter how small the probe that you can fabricate and how soft you can design them, as long as you want to implant those electronics into a mature brain, there is a significant mechanical damage that you can cause in the very beginning. So this makes us to think that if there's other way that can allow us to seamlessly integrate the nanoelectronics, especially those flexible nanoelectronics, into the whole tissue network without introduce those damage. So this is the direction that we spend a lot of time to study, that to really build the whole tissue interface without the mechanical damage. So we found that there is a solution actually from the biology, especially from the developmental biology. So you can see that the 3D brain is actually developed from a 2D cell sheet. So this cell sheet is generated from the embryonic stage that is called the neural plate. The neural plate will first fold into a neural tube, and this neural tube will ultimately fold into a 3D piece of the 3D brain structure. So if we can make a stretchable mesh nanoelectronics that is so soft that can be stretched by the tissue development, we may have a chance to integrate those electronics with the neural plate. And when the neural plate folds into the tube and ultimately 3D tissue, it will distribute the sensors across the entire 3D volume of the brain for this seamlessly integration. So we have this idea, and we start with the stem cell in vitro model to test if it is working. So in the stem cell development, especially in this recent organoids development, this 2D stem cell will first fold into a ball structure and ultimately fold into a 3D organoids, so that we can integrate a stretchable mesh nanoelectronics with this stem cell layer. And we expect that when this stem cell cell sheet fold into the 3D structure, it will distribute and fold this nanoelectronics across the 3D volume of the organoids. So this is a mesh electronics, stretchable mesh electronics that we design. We implement this serpentine structure developed by Professor Rogers' group into those mesh nanoelectronics. And this is this is the video of the mesh nanoelectronics. You can see that it's released from the silicon substrate, and it is so soft that even the floating of the water can easily stretch them. And we test their stability. They are very stable in the solution, so that we can integrate them with the stem cell sheet, and the cell sheet is start to fold into like a cell plate. And after 72 hours, they start to fold into this 3D structure. And after 20 days of the culture, ultimately the stem cell grow into a 3D tissue. You can see that we can distribute or embed those sensor network across the entire 3D volume of the organoids. And we call it a cyborg organoids for this technology. And using those embedded sensor, we can capture the multiplex electrical recording at the single cell, single spike resolution. And we can even capture the developmental process of those like electrical waveform. Those like waveform start with this slow calcium waveform, gradually emerge the hyperpolarization, and ultimately have this like a depolarization spike, which indicate like different ion channel expression from those cells. And we can even capture this like gradually synchronized cellular network from the initial like random firing to this like ultimately synchronized activity. We can induce those tissue into different kind of organoids. One is like a different like the brain organoids. We can capture this like an emergence of the electrical activity from this 3D organoids and keep recording them for as long as we want. This is an example that we record the brain organoids activity for almost like a half a year, and we can capture their like evolution of those like brain organoids over this time course of the recording, even to determine when the neurons in the brain organoids have already been changed from the immature state to a mature state. So the next step is that we will integrate those stretchable mesh nanoelectronics, not only on the organoids, but in a real like animals in the real embryo through the embryogenesis to implant the nanoelectronics and seamlessly integrate them with the brain tissue to form these cyborg animals. So we believe that in the very soon that we can build those bioelectronics that not only provide the long-term stable recording, but also record the neurons from the 3D volume of the brain tissue. So next to that, there is actually the third challenging for this bioelectronics tissue interface, which like previously was not captured, not well considered by the community of the bioelectronics. That is different neurons, is different cells, especially different neurons, will have the different cell types, the molecular defined cell types. For example, the different neurons will have a different gene expression, and also the different morphology and the connectivity. Collectively, they give rise to the different kind of electrical property. So we not only want to record the neural activity, but we also want to know which type of the neuron that we can record. So there is this third dimension on this diagram, that if we want to build this like a very effective brain or tissue machine interface, that is, we need to know the cell type specific information. So in the other field, like optogenetics, they already solved this problem. They solved this problem by harvesting this very unique genetically encoded molecules like channelopsin or hylodopsin, which is the ion channel, which can be specifically expressed on the specific type of the neurons to bridge those like optical information to those like genetically targeted neurons inside the tissue. However, for the electrical recording, there's no such interface that has been built up. So, but meanwhile, we know that there's those kind of conducting polymer, which can like substantially reduce the interfacial impedance between the implanted electronics to the tissue network. And we also have this very clean mesh electronics and the tissue interface. So that we think that can we also grow a layer of the bioelectronic interface from this specific type of the neuron within this intact 3D brain tissue to bridge the specific type of neuron with the electrode.
So this is we call the genetically targeted bioelectronics, that is through this in situ synthesis of the conducting polymer in the neural tissue as the interface. And what we are doing, that we do a lot of like a protein engineering. We screen the enzyme. We found a type of enzyme called APEX, which can enable this like in situ and in vivo polymerization of the different kind of polymers. And those enzymes, the type of protein, so that it can be specifically expressed in the target cell type in the brain, so that we engineer those enzymes, allow them to express close to the cell membrane, and then we introduce the conducting polymer precursors. It will cause the conducting polymer polymerization on the surface of the brain. So you can see that this is showing that the bright field imaging of the neuron of the obviously like for those neurons which express this APEX2, after we introduce the conducting polymer monomer, in this case the aniline, there is a very clear dark shell that is formed on the surface of the neuron. And this remarkable is that we can culture those neurons on the electrode array, and after the polymerization, after we dry this like a tissue network, obviously the neuron has already been died, but they are not very conductive except from those like cells which express those like enzyme and have this conducting polymer formed on them to build this like a detectable conducting pathway, bridge the individual electrode to the neuron. And of course, we can further extend this type of the work, not only for this kind of electrical recording, but for this kind of neural activity modulation. So if you look at this like equation, which is the Hutchinson-Hooksey model, very famous equation to determine the electrical property of the neuron, there's this very two components that is highlighted here. One is the membrane capacitance, another is the ion channel conductivity. Both of them like determine the intrinsic electrical property of the neuron. And for all those traditional methods like electrical stimulation or optogenetics, which really just manipulate the ion channel conductivity, open or close a specific ion channel to excite or inhibit neurons. There's no like a consideration about the membrane capacitance in this like a neural activity. So if we are not just like incorporate the conducting polymer, but we also incorporate the insulating polymer like a dielectric layer to the neuron plasma membrane, we can effectively tuning this like a membrane capacitance. Therefore, we can actually modulate the neuron intrinsic excitability, action potential propagation, or synaptic integration at the time scale that is much relevant to the brain, to the time scale of the brain function, and meanwhile in a long-term stable manner. So we have to do this. So this is a very beautiful work done by those like collaborations with Genetically and Carl Dessers when I was a postdoc at Stanford, that we use the patch clamp to patch the same neuron before and after the polymerization. And we found that for those like neurons have the conducting polymer polymerization, there's a statistically significant increase of the membrane capacitance. And meanwhile, for them to polymerize with another polymer, the DAB polymer, which is an insulating polymer, so there's a statistically significant reduction of the membrane capacitance. And if you look at this like a neuron spike recorded from the patch clamp before and after polymerization, you can see the significant reduce of the neural activity versus the increase in neural activity from this conducting polymer or insulating polymer polymerization. And we, of course, test this not only on the cell, but on the freely behaving animals. In this case, is on the C. elegans. You can see that we can target the polymerization onto one specific type of the neuron. In this case, the inhibitory motor neurons, so that after the polymerization, the C. elegans will not be able to go straight away, but like keep like going circles locally, which is exactly the same compared to the previous like a result that is done by the optogenetics to inhibit the inhibitory motor neurons. However, this is untethered and autonomic controlled technology, which can enable this long-term stable and manipulation or modulation of the neural activity. So this is the third part that we developed this genetically targeted bioelectronics to enable this cell type specific interface for the neural recording and the neural modulation. So together in the future, that we really want to combine all those technology together as one platform that we can enable this organ-wide long-term stable bioelectronic interface for the stable neural activity recording and modulation at the single cell resolution, millisecond temporal resolution, and with the cell type specificity. So in the end, I really want to thank all my students and postdoc in the lab. That is all those like work done by them, and also have a very fantastic collaborators from Harvard, MIT, and Broad Institute, and also want to thank the funding agency from the NSF, as well as the Harvard internal grant and some foundations. Thank you for your attention. Happy to answer any questions.
Well, thank you very much. This is really a wonderful talk. So exciting. Yeah, I heard some of the stories. I know that you present a lot of unpublished results. I heard some of the stories before, but today when I heard the stories again, I got excited again. So yeah, congratulations. This is extremely wonderful. Now, before so there's some questions asked from the audience, so I received them, and I will just mention those questions later on to John and also other people in the audience today. But before I approach that questions, I just want to say that Professor Jia Liu is probably the most visionary scientist I have seen, one of the most visionary scientists I have seen. And I have been very fortunate to have been working with him and having this connection with him for many, many years. So I still remember that multiple years ago that Jia mentioned a few things to me about his vision, his bigger pictures for bioelectronics field. I'm sorry. Some visions for bioelectronics field, and at that time I told Jia that, wow, this is the kind of challenge. It's quite risky, and I'm not sure how it could be implemented. Yeah, but what turned out that just in a few years, less than 3 years, and Jia and also his group have accomplished all those big things, really fantastic things, just in a very short period of time. So I'm very happy to see this happen. Now, Jia, I have some questions that are asked by the audience, and I will just ask them. Now, so one question is about the aging. So you showed this very beautiful recording, chronic recording, and covering mouse. I forgot it's mouse or rat, but it's like age from 5 months to 18 months. So one question is, how does the brain structure evolve over this time? So for example, would the brain shrink or over time? So how would that affect the structure of the mesh electronics?
Thank you very much, Boji, for this question, and thank you for your the introduction. I mean, that's definitely. I mean, you are also a very the most brilliant people I have ever done in my career. And so this I can show this slides. So the idea is that we can keep recording those activity over this long period of time, and we also do the characterization of the brain, and we can see that there's a sliding the shrink of the brain. But since the mesh electronics is completely embedded inside the brain tissue, it looks like they can still stably capture those neurons. We have done this research that is very carefully. We only consider that because for each electrode, there's multiple neurons that you can collect, and you basically use the spec sorting to isolate them from one to another. We are not looking at those neurons, that is those signals which one electrode only capture one neurons. So all those neural activities captured by the same electrode, we can see that there's one neuron
that is basically show a very stable like electrical property, basically suggests that this electrical probe to the brain tissue interface is not degrading over the time. And but this is but meanwhile, there's another neurons which show this kind of change of their electrical property over this time course of the aging. So that we think that this type of the change may be associated to the process of the aging. And meanwhile, we do this kind of like a machine learning like a data analysis, which is look at those like features of those like electrical waveform of the neurons, and show that there's like this like a change over time, which is so-called like a pseudo time that is already widely used in the genomics to look at the gene expression change over the time. And we compare this pseudo time to the real time, that which shows a remarkable like the overlapping of this trend over this time course of the recording, that it suggests that this kind of like electrical property change over time could be associated with the aging process. Yeah.
I see. Well, thank you, John. Now, the second question is I would say it's more like a comment. So, do you think, what's your perspective? So, you mentioned those cyborg organism, and I personally I like that concept as well, and I think this is really cool. And besides, like looking into animals, do you think there's also a possibility of focusing on plant? Like cyborg plant, right? Because the plant, yeah, their growth is right. It can be slower in terms of growth, and the motion is less. And if you can incorporate those flexible electronics on the plant surface during their development, then you can use the electronics for later monitoring. Right, that can potentially be developed into some very smart sensors that monitor chemicals or even other like simple things like temperature, humidity. That is relevant to the plants or any other things that plant growth would need. So, what do you think about that perspective?
Yeah, I think that would be a very interesting direction that we should explore. I mean, so we are mostly like focused on those like human tissue. That is mostly due to the like NIH grant requirement, because all the grant is like asking for the health related research. But I can imagine that integrate those like smart sensors, those tissue like nanoelectronics with the plant will not only help us to really understand like if there's also like a electrical property, electrical activity that govern this developmental process of the plant, but as well as for the potential for the future like agriculture related applications to help us to do this type of the in situ, also long-term stable recording of the plant during this like during their growth. Yeah, I think this is a remarkable idea that I definitely like very like willing to look at this direction and maybe give it a try in the near future. Yeah.
Yeah, that would be great. Now, the last question actually is come from my side. So I have a question about the genetically targeted chemical synthesis. As I introduced, when I introduced you, I mentioned that from my perspective, I really think that probably is the most exciting advance in neuroengineering, neuroscience, especially when combined with the materials. Now, my question is, do you think there's a way to make the material synthesis reversible? For example, initially, we can synthesize something on top of the surface of neurons that can be conducting polymer or insulator. But then after we pass certain time, right, we can remove those polymers. Is there a way to do that, making them reversible?
Yeah, so I think this is a very good question, and my group are very interested in this direction as well, and it is quite critical for this type of the technology to be used in, especially in the clinical application, that it need to be like removable. So one direction is that those conducting polymers will like gradually disintegrated
inside the biological system. It's like that's why the PDOT cannot be used for the very long-term stable recording, because it's naturally like integrated over the time, although it cannot be like degraded. And this integration, after the integration, what's those like polymers and materials impact on the the tissue healthiness is still not 100% clear. So what we want to do is to introduce those like very specific designed molecular structure into those conducting polymer monomers, which have been already been studied widely previously by a lot of people, include like Genetically at Stanford, Genetical at Stanford. That, for example, the amine group that integration into those like the dimers of the aniline after the polymerization, over the time they can quickly like degrade inside the biological system. So we want to provide this long-term stable modulation on the neural system, but meanwhile we can provide this like kind of a gradually degradable polymeric materials inside the tissue. Yeah.
I see. Well, thank you, John. I think those are all the questions we have for you today. And any other things I just want to ask to the audience before we close this? Okay, all right. So, well, thank you again, John. So maybe I think yes. Hi, Pierre. So you can finish this webinar.
Thank you very much, Bozi, for sharing this webinar today, and thank you very much, Professor Liu, for such an interesting talk. I would like to thank you all for attending today's webinar and for your questions. If we didn't manage to get back to you on any of your questions, please apologize. We are actually very close to the end of this webinar. I hope you found the talk and discussion useful, and I hope to see another of our upcoming webinars. Have a good rest of the week, and see you later. Bye.
Bye-bye. Thank you.
Bye, everyone.